Okay, good morning, good afternoon, or good evening. I'd like to welcome you to this webinar, Streamline Your CLL Analysis and Alleviate the Burden of Running Multiple Assays, introducing OGT's SureSeq CLL and CNV NGS panel. The presentations will be delivered by two of my colleagues at OGT, Kai Brands and James Reed, and also Flecher Fortam, Scientific Director of the Cytogenetics Lab within the Medical Division of Sherbrooke University Health Centre in Quebec in Canada. My name is Dave Cook, Senior Manager of Regional Marketing at OGT, and I'll be your host for today's webinar. Before we could begin the presentations, we just have a couple of housekeeping points to address. To type in a question at any time during the webinar, you can do this by clicking on the Q&A window on the top right hand side of your screen, and you can just type in your questions here. All attendees will be in listen only mode to avoid any background noise during the presentations. So if you are experiencing any technical issues, then please contact us via the chat box. The chat box can be accessed by clicking on the speech bubble icon on the bottom right hand corner of your screen, where you can send your message to the organizers. With that, I'd like to introduce our first speaker, Kai Brands. Kai has been working in the next generation sequencing field for almost a decade, both in a research and commercial setting. Kai is responsible for OGT's oncology NGS portfolio. Over to you, Kai.
Thanks for the introduction, Dave. Hi, my name's Kai and I'm responsible for the SureSeq range at OGT. Today we'll be focusing on the SureSeq CLL plus CNV panel, which we launched earlier this year. This panel is capable of targeting both SNVs and indels, as well as a wide range of CNVs implicated in CLL. And with that, I'm going to switch to my slide and start my presentation.
I'm sure most of you are very familiar with CLL, and Flecher, our next speaker of today, will have some further details as well. So I'm going to keep this introduction brief. CLL is the most common leukemia in the Western world and accounts for roughly 25 to 30% of leukemia cases in the US. Boiling down to an estimated total of just over 20,000 new cases in 2019 in the US alone. Genetically, CLL is a heterogeneous disease, and a wide variety of chromosomal abnormalities are associated with CLL, ranging from single nucleotide variants, SNVs, insertions, deletions, indels, up to large copy number variations, CNVs, and these include trisomies as well. Research into aberrations associated with CLL is crucial to characterize risk and treatment options. Some examples include 17p deletions covering tumor suppressor gene TP53, as well as 13q deletions and trisomy 12. Next to these CNVs, over the last few years, our genetic knowledge of CLL has grown significantly. And next to TP53, research into more genes and mutations has identified specific genes to be of prognostic value or serve as predictive markers for example, therapeutic responses. If we focus on CNVs alone, we can see how important it is to investigate CNVs in CLL, as they're very common, being found in over 80% of CLL cases. The most common CNVs are deletions in 13q, covering the RB1 DLEU genes, including DLEU2, which harbors a microarray cluster 15A16, important in CLL as well. The other four CNVs found in CLL completing the top 5 of most common CNVs in order of frequency are 11q deletions covering ATM, trisomy 12, 70p deletions covering TP53, and 6q deletions covering MYB. As mentioned on the previous slide, next to these CNVs, research into recurrent mutations is important to identify genes with prognostic implications such as Notch1, SF3B1, MYD88, and BERK3. Next to these, an example of 2 genes with mutations being associated with therapy resistance include BTK and PLCG2. So translating this information into CLL testing, we see that it's imperative to investigate both structural aberrations as well as SNVs and indels in a variety of genes. To address this and obtain a comprehensive genetic picture of CLL, researchers often employ testing strategies involving multiple different methods. Depending on the number of genes under investigation, Sanger sequencing or NGS are used to look at small variations including SNVs and indels, while techniques such as FISH, karyotyping, and microarrays are used to address structural variations. As an example, labs only looking at TP53 mutations can choose to do Sanger sequencing and one of FISH, microarray, or carrier typing for investigation in deletions in 17p. Labs who have transitioned to NGS can use NGS to look at TP53 and benefit from the higher sensitivity of NGS compared to Sanger, and can design larger panels to look at some of the other genes we discussed in the previous slides as well. However, they would still have to employ a secondary technique to investigate structural aberrations. So to alleviate this burden of running multiple assays and to help labs further their research into CLL, we collaborated with recognized experts in the CLL field and developed the SureSeq CLL plus CNV panel. So what does this panel deliver? The CLL plus CNV panel, like all our SureSeq panels, benefits from OGT's long history and expertise in hybridization, which runs through the microarray and FISH products as well. The expert probe design and hybridization-based panels allow for unparalleled coverage uniformity, allowing variant calling of low-frequency SNVs and indels, even in traditional difficult-to-target genes, for example, genes rich in GC content. However, what really sets apart the CLL plus CNV panel is the CNV detection, allowing accurate CNV calling ranging from loss of single exons up to complete chromosome arms and trisomy 12. Combining CNV detection and SNV indel calling in a single NGS assay will allow you to streamline your laboratory workflow and cut down on turnaround time. Just like all our other SureSeq panels, the SureSeq CLL plus CNV panel comes with our complementary NGS analysis software called Interpret. I'm not going into Interpret in great detail in my presentation, but James from our computational team will focus on our easy-to-use NGS solution later. Now let's have a look at some of the details. The CLL plus CNV panel targets a total of 12 genes implicated in CLL, including the genes I mentioned in the previous slide and a handful of genes which you can see on the screen here. In all these genes, we can detect SNVs and indels down to 1% variant allele frequency. Then for the CNVs, the panel targets the 5 most common regions known to harbor CNVs in CLL, which as mentioned before are 17p, 11q, 13q, 6q, and trisomy 12. And for the first 3, we can also detect loss of heterozygosity. Detection size ranges from single exons in the targeted genes in these regions up to the complete chromosome arms and trisomy 12. In terms of detection limit, we're able to call CNVs down to 10% variant allele frequency, equating to 20% tumor content. The panel includes 2 genes and a 24 SNP profile for sample tracking, has a total size of 117 kb, and this allows for pooling of up to 16 samples on a single MiSeq v2 sequencing run. Now let's have a look at some of the data generated with the CLL plus CNV panel. First of all, focusing on the SNV and indel detection. In the 2 examples here, the top example shows an exon 15 hotspot variant in SF3B1 with a frequency just below 5%. You can see the software has flagged the variant allele in the window here. At the bottom, we see an exon 4 frame shift deletion in TP53 with a frequency just below 40%. In the coverage window, the reduced coverage indicating the deletion is clearly visible as well. You can see the panel in combination with our Interpret software nicely picks up these 2 variants and is able to do that down to 1% allele frequency. Then going into the CNV detection. Shown here is a 42.7 MB deletion on 11q covering ATM. You can clearly see the vertical zone of higher probe density in the ATM region in the example. Importantly, these samples were all compared to array data as well, and you can see that the NGS results are fully concordant with the array data on the right. Interpret provides tumor content estimations as well, and this example has an estimated tumor content of 30%. Moving on to a bit of a more complex deletion, which is again picked up by the panel without problems and fully in line with array data, as you can see on the right. The figure shows a biallelic deletion of 0.6 MB within a larger single allele deletion of roughly 1 MB in the region covering the DLEU genes on chromosome 13q. You can see that even the small deleted region on the left, equating to 80 kilobases, is picked up accurately. For my final example here, I'm showing a trisomy 12 with an estimated tumor content of 47%. You can appreciate the reliable gain call across the chromosome Again, in full agreement with the array results. Flechette will be showing some further data examples in her presentation. So for my last few minutes, I just want to briefly introduce our NGS analysis solution, Interpret. As James will go over this in a lot more detail, I'm just going to give a short introduction at this point. Interpret is complementary with all SureSeq panels, including custom panels. Interpret is designed to detect both SNVs and indels, as well as the CNVs we discussed today, and is actually capable of detecting a wider range of structural aberrations, including translocations, for our other panels targeting translocations as well. Interpret is highly customizable with multiple QC metrics, a comprehensive range of filtering options, and has a built-in IGV window. Interpret can link out to multiple variant annotation databases, generate tailor-made reports with a wide range of reporting options, including variant and QC reports. For security and control, Interpret has the option of giving users different access permission rights, offers logging and tracking of users, and also allows for the easy porting and backing up of all data. And with that, I've come to the end of my presentation, and we'll go back to you, Dave.
Many thanks, Kai. I'd now like to introduce our next speaker, Dr. Flechette Fortin. Flechette began her scientific career as a lab technologist in cytogenetics in 1997. After finishing her PhD, she completed a clinical fellowship in cytogenetics at the Canadian College of Medical Genetics. Dr. Fortin is now Scientific Director of the Cytogenetics Lab within the Medical Division of Sherbrooke University Health Centre in Quebec, Canada. Recorded at the recent Cancer Genomics Consortium virtual meeting, Dr. Fortin will outline the laboratory's transition from OGT cytosure hematological cancer and SNP microarray to the SureSeq CLL and CNV NGS panel utilizing OGT's Interpret software. Dr. Fortin will discuss the strong data concordance between the two technologies and will be available for the Q&A session at the end of the webinar.
Today I'm going to discuss the genomic evaluation of chronic lymphocytic leukemia and how our lab decided to transition from an aCGH SNP platform to a targeted NGS-CNV detection approach. So, first, I have no conflicts of interest to disclose. As you well know, CLL is the most frequent adult leukemia in our populations, with an annual incidence of 2 to 6 cases per 100,000 people. It can manifest in a ganglionic form, SLL, but most cases present as a leukemia characterized by a progressive increase of a monoclonal lymphocytosis. Newly diagnosed or treatment-naive patients are classified using the CLL IPI, which combines both clinical characteristics divided in 5 rise stages. And also it combines genetic characteristics. As you can see, only TP53 alterations and IGVH mutation status have a specific value in the CLL IPI. These characteristics define 4 risk levels with distinct overall and median survival and will lead to the ultimate clinical decision: to treat or not to treat the patient. On the other hand, in their recent clinical practice guidelines, NCCN gives more room to genetic factors and will propose therapeutic avenues according to the findings. If there's no treatment indication, the clinician will need to wait and observe the patient. If or when there's a treatment indication, or at relapse or treatment failure, then you must perform a complete genetic evaluation, which will include a FISH genetic evaluation with 4 targets, TP53 sequencing, a karyotype to assess the complexity of the disease, and also aCGH mutation status. Since more than 20 years now, FISH is the gold standard to perform genetic evaluation of CLL. 5 different types of results can be distinguished by targeting 4 chromosomal regions. both deletion 17p and 11q are associated with unfavorable prognosis. In a survey of fish results issued by our lab in 2015 and 16, just before our transition to aCGH, 24% of cases had normal fish results. As you can see, 40% had an isolated deletion 13q, while 12 to 13% patients were having either a deletion 17p, a deletion 11q, or a trisomy 12. Even though some hematologists are reassured by a favorable result, targeting these 4 regions is costly, and many labs face budget restrictions. So the combination probe ATM TP53 becomes appealing since you're using only 1 probe, 1 slide, with 2 targets. However, at the end of the day, FISH still has its limits and is unable to detect complex karyotype or multiple clones, with a few exceptions. With the advent of synthetic mitogens that replaced pokeweed as a, we must say, poor B-cell mitogen, CLL karyotyping has gained new adepts. Complex karyotypes associated with an advanced disease and unfavorable prognosis can be observed, as well as multiple trisomies accompanying trisomy 12. However, it doesn't replace FISH, which means more labor into the lab. The quality of chromosomes obtained is not the best, and karyotyping remains difficult for CLL. In an effort to simplify the workflow into the lab and gain more insights into the genomic profile of CLL, aCGH can be used to replace FISH with a turnaround time similar to karyotype in our lab. In fact, we have a small lab. The analysis is quite simple, well, most of the time. And less hands-on time is required since these onco arrays are labeled at the same time as our constitutional arrays. The ability to perform an whole genome analysis with aCGH is a plus, as is visualization of genomic complexity. However, for a subset of clinicians, this can be viewed as too much unnecessary information when first treatment decision comes. aCGH remains a costly analysis, but, in my opinion, not significantly more than a full FISH analysis or karyotyping. However, mutations are not evaluated and need to be done separately. Actually, our center performs a genomic evaluation of CLL using aCGH, and a sent-out is made for IGHV mutational status. For most cases, either at diagnosis or at therapy failure, TP53 sequencing is not done. Only occasionally, TP53 is sent out for sequencing. Thus, we would like to give all our patients an easy, automatic access to TP53 mutation analysis. Then, 2 years ago, when we were presented by our OGT representative an NGS test that would permit both gene sequencing and targeted CNV detection for CLL, we were a bit doubtful. On paper, it looked great and promising. It would detect only clinically relevant CNVs as per NCCN guidelines, and would detect somatic variants in a lot of genes, including TP53. Thus, 2 of our current clinical concerns would be fulfilled: have an easy access to TP53 sequencing, and give targeted CNV results for treatment-naive CLLs. However, would an NGS assay based on capture really be able to detect accurately large CNVs as observed in CLL? We were really not sure. And then we were offered to participate in a product development trial, and we said, sure, yes, we're interested. So we sent out 32 DNA samples to OGT in UK from patients already characterized by aCGH. All tissue and aberration types were present. Of course, I included some tricky samples to push the test at its limit. I must admit that when the results came out and were presented to us a few months after, we were indeed impressed. In the next slides, I'm going to show you some of these samples with side-to-side CNVs detected by aCGH and by NGS. To begin, an easy one, a classic trisomy 12, easily detected with aCGH. In fact, easily detected with NGLs also. Even though the base coverture is not as dense as in CGH, this old chromosome CNV is readily detected. Then a complex case, number 18, having deletions 13q and 17p in all cells, and a mosaic deletion 11q. Here, a nice type 1 deletion 13q with a good detection by NGS, and as you can see on the gene track, centered around DELU1 and DELU7. And then the deletion 17p, which extends past the centromere on aCGH profile. As you can see, the NGS coverture extends that far too. I'm very happy to see that. And for this case, we were also having a deletion 11q in mosaic. With this one, I wanted to know how extended was the coverage along the 11q arm. By CGH, the ATM-centered deletion spans 28.4 megabase. And with the detection by NGS, this deletion extends at 27.9 megabase. And the mosaic, well, it's really well detected, and the estimation is quite similar. I'm happy with that. Now let's continue with case 21, another tricky one, where we can see a mosaic deletion 13q and also a deletion 11q, which is biallelic. By aCGH, I had manually flagged this deletion 13q on the CGH profile, so I was expecting that the NGS could see it automatically. And yes, it's true. In fact, the NGS really detected it, and it's nicely surrounded, as you can see. The estimated tumor content is also similar, which I am happy to. Then for case 21, the deletion 11q, which is biallelic, with two different sizes. Again, I'm happy. Both can be observed on aCGH or by NGS by looking at the profile. I can delimitate the smaller deletion on the aCGH profile easily. As for the NGS, I can import the data on my CGH software with a mouse click and do the same. This is good. As for tumor estimation, it will be better evaluated after redesigning the aberrations, as usual. Okay, another biallelic deletion on chromosome 13 for case 26. Very happy. It's well circumscribed by NGS, as you can see. And I keep the most difficult one for the end. A deletion 13q, biallelic, with the same size of the deletion on both chromosomes. How would it be interpreted by the software, since almost no reads were generated by NGS? Wow, now I'm very impressed. The software did catch up a biallelic deletion was present. And see the log2 ratio. It's around -3. It's very low. And it even called separately this small monoallelic deletion here. I'm very happy. So, what's coming up next in our lab? We are starting our clinical validation with this summer. Again, all tissue types, all aberration types, including LOHs, are evaluated. We would like to perform a true dilution study with the patient's normal DNA side to side with its CLL DNA. And we want to do it so since the SNP sequenced by NGS are important for correct CNV detection. We'll try to obtain a number of buccal swabs from 2 to 3 selected patients with both trisomy 12 and a deletion. We'll first confirm that buccal swab DNA is normal by running the array. If so, yes, we will be able to do this dilution study. When CNV will be validated, we will launch the test and then start SNV validation. A new testing algorithm will be implanted in our center since relapse and refractory samples will have both an aCGH and the NGS assay. So we will need this specific information from our clinicians. I do expect some difficulties on this. As you know, test requests can have very low detail levels. We think and hope that our clinicians will find it useful to have an easy access to TP53 sequencing data, as well as other relevant genes mentioned in the recent evidence-based review published by CGC. All but one gene mentioned as unfavorable prognosis in this paper are sequenced in OGT's SureSeq CLL plus CNV NGS panel. With this, I conclude my talk and will be happy to answer your questions.
I'd now like to introduce our final speaker, James Reed. James has been working within the computational biology group at OGT for 7 years and leads the data analysis team who developed the NGS pipeline used in the Interpret software. Over to you, James.
Thank you, Dave. Hi, I'm James, and welcome. I shall now give you a very brief demonstration of the Interpret software. I'll switch to the screen. The NGS analysis software Interpret is a web server that can be accessed using any web browser. Past the login prompt, user is greeted with this homepage with the different menu bars at the top and the options here to the right, which I'll describe in more detail later on. At the bottom there are the 3 simple steps in the workflow we propose, which is for the user to upload their FASTQs, run a batch of FASTQs with specified parameters, and then once that has completed, the user can view the results of that batch. So we'll run through these options one by one. So the upload FASTQ option allows the user to drag and drop some FASTQs, any number, or actually specify the full path. Once these have been uploaded to the system, Interpret can extract the sample in automatic, a sample ID from these FASTQ IDs, and the user can, if they want to, override this this option. Once the user is happy with the sample IDs, they can upload the rest of the FASTQs. That will be run in the background, and whilst the upload is ongoing, the user could do other operations within the software. Once a set of FASTQs have been uploaded, the user will be prompted to run a batch using these FASTQs. The recently uploaded FASTQs would be listed here. Here I'm just showing you a few. And at this stage, there is essentially 3 pieces of information that we have to provide to run a batch or a set of FASTQs, a run, for example, from the sequencer. The first one is the easiest one. It's the name of the batch. And the next one is the panel with which the data was generated, so which set of baits were used. In our case, since we're showcasing the CLL CNV one, I'll be using the SureSeq CLL CNV panel. This, in essence, is a full specification of the design of the panel. So it goes beyond the so-called bed file of genomic coordinates of the targeted regions. It also includes information related to our targets, which help the analysis of the different variants to be identified during the analysis. The last part of this batch creation is the selection of protocol. And this, here we have, for example, a default CLL CNV protocol. I will show you next how this is defined. But this essentially is a list or a recipe, if you wish, of parameters that will be used during the analysis. So these parameters will be which type of variants we want to identify, what parameters to use, depending on which parameters we are looking for, and finally which type of cutoffs to use. And then once the results have been obtained, which filtering should be applied to the end results. Once these three options have been put in place, we would just be clicking down here at the right hand side and running an analysis. This analysis as well would run in the background, and we could do other operations in the software while that was happening. Now I'd like to describe a little bit more detail the protocol. The administrator of the system can define a protocol in the Analysis tab. Here we are listing the different protocols on the system, and you can see that the CLL CNV protocol is currently version number 3. Each of these versions is stored in the backend database, and the user can see what has changed as the versions evolved. It has a default metric set associated to this, so these are quality control parameters or boundaries to specify labels for each of the quality control parameters. It's easier if I show you briefly what this consists of. Here's the default metric set. As an example, we've set boundaries as poor, good and excellent for the percentage of reads aligned as less than 80%, between 80% and 95%, and then above 95%. So we can do that for a number of quality control metrics. The user can choose which ones to include and which boundaries to use. This enables the user to identify potentially problematic samples in the run, or indeed samples that do not meet the specified criteria. It tells us who created it and when, and then we have a set of filters that we'd like to apply when using this protocol. So these would be default filters that would be applied during the generation of the results, and so the end user would only see the resulting filtered set of results and not all of them. In our case here, just for demonstration purposes, we're leaving all variants to come through. And then down below here we specify which variants to identify. We might wish to not do any CNV or LOH calling and so forth. This is also dictated by the type of panel that you'd be using. And then in the advanced pipeline configuration, we can actually be much more precise and go and change particular parameters for each of the different variant types, so such as SNVs and CNVs. And then finally, for each variant type, the user can specify different cutoffs of detection. Once the protocol has been saved and defined and saved, then the batch can be run, and once it's completed, the end user would go to the batches view and see the completed batch. So this view represents the batch view, where you have the results for each sample, and here at the top we have an overview of the batch, so a brief overview of who ran it and when, which panel was used, the actual status of the batch, if it's completed or in progress, which protocol was used, so this is similar to the one I was showing you earlier, And then finally we have what we call a multi-QC report. This is a separate web page that encapsulates all of the quality metrics for all of these samples in this batch. And it's interactive in the sense that the user can filter, sort, export, and so forth, these different filters. We have customized plots that we've incorporated into this report to enable the swift identification of any potential problem during data generation. Back to the batches view, we have here a number of buttons. So there's a refresh button. There's a file status button which enables you to download the results for particular samples. So results in terms of, for example, BAM files such as the aligned reads, QC result files, or the VCF files with the variants and so forth. These can also be downloaded as a bulk, as one single compressed archive. We have the copy batch ability, so this simply copies over all of those FASTQs that were used in this batch and can be reprocessed, possibly with a different protocol. For example, if the batch is running and some problem has been identified, the end user can stop this batch. This is this button here. And then finally there is a generate report button where the user can specify which type of report to generate for this batch in its entirety. So this would comprise the quality control metrics and indeed all of the variants that have been identified. And these can be completely customized by the end user. So down below here we have for each row the results that were obtained for each sample. We can see the number of variants that have been identified, and then we have a number of quality control metrics that we can add. Here you can see easily customized, can be easily customized by adding these to your view, and you see these are added, and you'll see that they've been color coded, and this color code reflects the specifications that were detailed in the metric set in the protocol. And so this is telling us that highlighting us possibly problematic samples or maybe issues in the underlying data. And if there are such issues, the user can also go and zoom in into individual quality control reports for each sample. Now to the results. If we could, for example, we could select one sample or multiple samples, and then we'll start with CNVs, for example. Here we have at the top the result of the filter from the protocol that was applied to the results. So in our case we had nothing, so 8 CNVs and LOHs entered and 8 came up. Here we have the actual results, so in terms of CNVs and LOHs, we might want to say focus only on CNVs. And within the table of variants here, you can see those different variant attributes that are generic coordinates and so forth. We can click on this one on chromosome 13. You can see that it's already been colored red, and that's because previously in the system this CNV has been classified as pathogenic. This is an option that we have here. If we right click the actual entry in the table, we can add it to a shortlist. We could clear the classification, remove it completely from the system, override it, maybe decide that we're not completely sure about its status in terms of it being pathogenic or similar. And in addition, this kind of data can also be viewed in our aCGH cytosol interpret software if there's a local installation. That enables you to match up maybe previous data that was generated in the lab, and finally we can export and go and see this particular CNV in different external databases. In the track down below you have an IGV browser view, but instead of the standard SNV view of your BAM files, here we are highlighting log2 ratios in a very similar manner to erase CGH data. So where these log2 ratios here deviating from the 0, which would be a copied neutral area, and here they're negative and highlighting a deletion. And I'd like to quickly zoom in on here. I don't know if it's going to be possible to view, but you'll see the red dots. For each red dot there is a grey dot just above it, and that grey dot is the original data, and this red dot is the corrected data. And this is something quite innovative, and we've implemented by estimating the tumor content. So in this case we said there's 87% tumor content, and adjusting the log2 ratios according to this tumor content to enable better CNV calling. And this tumor content is estimated using the LOH frequencies that we can observe in the data across the panel. This LOH track is displayed down below, and the end user can also add different, speaking of tracks, here we have a managed track facility. So here you could add, for example, different tracks as long as they're in bed format. And finally, the user could also, at this stage, just as you can at the batch view, you could also simply generate a report here of the variants that have been identified so far. So that was for CNVs and LOHs. We could also have a look at SNVs. So this is more of a more traditional, let's say, NGS view with IGV. I'll just click on the first entry here to show you the actual coverage track here. What the software is doing is loading the BAM file. And here you have your pile-up at that particular position, and a normal IGV view sort of operation could be, for example, sort by base, and then you can see all of your Gs lined up there. Here the concept is the same. We have a filter that was applied at the protocol level. We could apply one here because maybe we want to exclude certain variants. So if I show you a default SNV we have created earlier, and this is a combination of these logical operations, we can use that filter and update it to the current view. Now this tells us that we have 30 SNVs to start with. We applied the filter, this was the filter we've just applied, and we ended up with 20. So we can zoom in on our variants of interest. And of course, to zoom in on variants of interest, we can add here, again, we can customize our columns, and we can see here that in addition to the attributes of the variants, there are a multitude of entries related to the pathogenicity of the variant. So for example, polyphenol SIFT scores. We could add minor allele frequencies from population databases, and also in particular we could add the impact of the variant, which I believe we already have here. We have the impact, the most severe consequence, or the significance that's been taken from ClinVar. So here we have one, for example, that's also been associated to a drug response. Similar to what I was showing you in the CNV view, the user can select the variant and classify it. Here in addition we have the ACMG guidelines classification and manual classification, and we could say this is abstract, there's maybe a benign variant, and we can use again external resources to help our interpretation of the variants. So we can also add it to a short list and so forth. This view is, well, for P53 is quite complex. There are many transcripts in P53, but this option enables the user to set a preferred transcript when viewing variants. Now, this can be done prior to the analysis, but also during the analysis, that we might say, no, we only want to see this particular variant. So we'd like to use this as a preferred transcript for P53. Say no. And that concludes the view of the batch view. I just briefly wanted to show you that we all, at the similar level, we have a samples view. So now we have the samples that have been processed, and we can see all the information for each sample in turn. The variants that have been identified. So we have a sample view and a variance view. And the variants we can see, we can track the different variants over the different samples from different batches. And this, again, we use similar tools if we want to see, for example, this particular sample. This would load in the IGD viewer down below. And with that, I will conclude my brief demonstration of the Interpret software. I hope you enjoyed it, and if you would like to have a more in-depth demonstration, we could easily arrange that for you. Thank you. Over to you, Dave.
Many thanks, James. Before we start the Q&A session, I'd just like to draw your attention to another short poll, which would be great if you could complete. It will just take a few seconds. Now on to the Q&A. We've been checking the actual sort of a box this afternoon as a few questions have come in. First question, I presume probably for Kai actually. Kai, what's the actual detection limit for CNVs in terms of size?
Yeah, thanks, Dave. So the CNV detection differs a bit per region. So as you've heard just now, so that we detect these 5 different regions on 5 different chromosomes. We're targeting for CNVs, and within the genes, within these regions, we can basically detect CNVs down to the exon level or up to the whole gene. And these genes, for example, include ATM on 11q and TP53 on chromosome 17p. Then for the chromosomal arms, we can detect roughly down to 5 to 10 MB range. And then for chromosome 13q, it's a little bit different. So we can detect type 1 and type 2 deletions, which are occurring around the DLU and the RB1 genes. And as shown in one of the examples that I had, actually within these regions, we showed just an 80 kb deletion. So we can go down quite deeply into those regions.
Okay. Thanks very much indeed, Kai. I suppose a similar question, Flesher.
Yes.
A question for you, probably similar to that as actually, probably from the work you've been doing. What's the actual sort of a lowest percentage for SNVs and indels where you're able to detect?
Actually, we haven't started to evaluate the SNVs and indels, only the CNVs. So
okay.
I'm sorry, I cannot answer.
No problem at all. So what did you find then in terms of actual detection limits, you know, in your hands?
In the CNVs,
yeah.
In fact, in my presentation, I really did put the most trickiest one. Let me see. Well, at this time, we have a 27% tumor content that has been readily detected. It was a deletion 11q, but we want in our validation to push the system at its limit and go as low as 10%. So pretty similar to what we would observe in fish.
Okay.
Since 20% alleles, well, would be 20% of cells, and it's pretty clinically significant.
Okay. Another question. Again, for Flesher, sort of, what sort of DNA inputs were you using in your work?
Actually, we have no problem with the DNA quantity that we have. So we use the highest number recommended, which is in 1 microgram.
1 microgram, okay.
So usually when we extract, we have something like 10 to 25 micrograms from the sample.
I'm presuming that with blood being the source, obviously DNA quantity is not an issue under normal circumstances.
Yes, in fact.
Okay. Right. question for James, actually. Can Interpret be integrated in our current software environment? I'm presuming they mean probably an upstream limb system. I suppose it could be the downstream variant annotation tool, but yeah.
I see. Yeah. Thanks, Dave. Yeah, I think I understand the question. Well, to a certain extent, certainly, yes. We have developed in the past plugins to accommodate such requests from the customer's side. So, as you said, you could have something upstream. So there could be a laboratory information management system with, say, sample information, sample tracking, and so forth. If we were given, you know, we'd have to know how to access that, but then that could be done, and then that information could be linked to the sample IDs of our sequencing runs within Interpret, and so that is a full integration, let's say. And then you mentioned variant annotation. So downstream, as I demoed, you are capable of downloading a lot of the results files. And of course, an important one there from the variant point of view is the so-called VCF file, which contains the variant calling results. And that could be used as input to an external third-party tool for doing variant annotation. So very much yes on that front.
Okay, thanks, James. A question came in a couple of minutes ago. For Flechair, what's the minimum percentage of plasma cells to maintain the same sensitivity as fish? Is that actually?
Actually, it's something that I don't know. Usually, most patients with CLL will have a big involvement into the blood, but I think that when we have a lower tumor content, it's probably because there is not that a high number of CLL cells present. But truly, I just don't know. We have never made a correlation between what we were seeing and what was the number of cells involved into the blood.
Okay. Kai, can the actual panel, this is actually linked to that first part of the question actually, can the panel actually detect sort of a IGV hypermutation?
So yeah, we do appreciate this region is of great importance for CLL, but this panel does not target IGHV region, so does not detect IGHV hypermutations. So I would say this is definitely something, definitely a region of interest to us. But at the moment, we don't offer a solution for capturing IGHV hypermutations.
Okay, right.
In fact, for what I know, the technique used to detect the IGHV mutation is not like detecting a CNV. It's much more by seeing if there is a lot of recombination in the region, so which will make the hypermutation of IGHV. So it's really not the same kind of mutation. You know, we use the same term, but it's not biologically the same. So it's technically not the same way to obtain the result.
Okay. One question here, actually, again, it's probably sort of from your experience, Flechair, what's the minimum tumor content needed to successfully process and analyze a sample?
As I was saying before, actually it's something that we don't know. During the validation, we will make a dilution study to get as low as 10% of an aberration to see if it's still detected. So it's still an unresolved question.
Okay. Right. I'm just looking at the clock now as well. I think, as we're approaching the very top of the hour now, I think we've now reached the end of our webinar. I'd like to thank our speakers, Kai, Flechair, and James, and to all of you for joining the webinar today. If we haven't had time to get to your question, we will look to get an answer to you after the webinar. And finally, if you missed any part of the webinar or wish to listen to it again, a link to an archived version will be emailed to all registrants. With that, very many thanks for joining this OGT webinar today. Have a good day.